Hello students, welcome to medicoRAS. Today we are going to discuss the anatomy of flowering plant in one go. So anatomy means the internal structure of any living organism. So here as we are discussing the flowering plants, that means we are discussing about the angiosperms. Now for the internal structure we must know their organs or organ system. As we know, the organ systems are made up of tissue. Tissues are the group of cells which have common origin and most of them are actually performing the same function. Now let's see types of tissue. We have two different types of tissue according to NCERT textbook. One is known as meristematic tissue and other one is known as permanent tissue. But sometimes in entrance examination, you may get another type of tissue and this is known as secretory tissue. The characteristic feature of meristematic tissues are: number one, the term meristose is actually represent the divided cells. Again, if we check, it means the ability of division. This cells has a special character that it has dense cytoplasm, big nucleus and small vacuole, whereas under permanent tissue, cells originates from both primary and secondary meristems. Again, they lost the ability of division and have some specialized structure and function. For the secretory tissues, we can write these cells are the dead cells and only can store substances like oil or other chemicals. Now, if we check the meristematic tissue, that meristem is of three different types: apical meristem and intercalary meristem. These two meristems are known as primary meristem. The lateral meristem, it is known as secondary meristem. Now, primary meristem develops in early age of the plant, whereas secondary meristem is responsible to increase the diameter of the plant. Under permanent tissue, we have two different types of tissue. One is simple tissue and other one is complex tissue. If it is permanent, then under the simple tissue, we have parenchyma, collenchyma and sclerenchyma. And if it is complex, then we have xylem and phloem. Similarly, if it is the secretory tissue, then we have the glandular tissue and the lenticephorous tissue. And again, glandular tissue can be divided into two types, that is the secretory cell and the glands, whereas lenticephorous tissues are divided into two types, that is latex cell and latex vessel. Secretory tissue can release oil, raisins, gums. Along with this, they can also release some digestive juices. Now, let's see what is the function of apical, intercalary and lateral meristem. So, first of all, under apical meristem, two distinct areas we have: shoot apex and root apex. Under the shoot apex, we have a concept of axillary bud. These are known as left behind cells when development of leaf and stem takes place. That is, if we consider this as a stem and from the stem, if one leaf arises like this, in between these two, we have an angle and this angle name is the axillary angle. So, the bud which is developed from this side or this angle is known as axillary bud. The axillary bud is responsible to give rise to either flower or branches of the plant. Under the root apex concept, we can see here the root caps are present just below the region of meristematic activity. This is the region of division, and the other region of the root apex are region of maturation where root hairs are also present and region of elongation. If we check the structure of the shoot apex and root apex, we can see this kind of structures. Like here the leaf primordia is present, which is ultimately going to give rise to one mature leaf. Whereas this is the main branch. In this we have vascular tissues and this is the leaf primordia. Then in between these two we have a specialized cells and these cells are nothing but the axillary bud. This axillary bud ultimately going to give rise to flower or the branch that already we have discussed. Similarly, here in this case we have one central cylinder which ultimately going to give rise to the vascular bundle, and near the sides of the vascular bundle we have some distinct areas is for the cortical region or the cortex, and surrounded this we have another specific area and is known as protoderm. Let me tell you, the protoderm is actually responsible to give rise to the epidermis in case of stem and epiblemma in case of root. Again, we have some initials of central cylinder and the cortex. That means from this particular zone only we can get initial cells for the central cylinder, or this particular zone only can give rise to the cortex. Below to this area we have some root cap initials as well, and this root cap initials ultimately going to give rise to root caps, and this particular zone is responsible for the root apical meristem. Now let's check the functions of apical meristem. The primary apical meristem is actually developed from the promeristem and it has three different types. First one is known as protoderm, next one is known as ground meristem, and the third one is known as procambium. Now from the protoderm, as you can see here, we will develop epidermis in case of shoot system, and if it is root, then the same thing we can consider it as epiblemma. Similarly, if it is ground meristem, it will give rise to hypodermis, cortex, endodermis, pericycle, medullary rays, whereas procambium only give rise to the vascular bundles. Now, the first layer or the epidermis give epidermal tissue system, which we also considered as dermal tissue system. Ground meristem, which includes hypodermis, cortex, endodermis, pericycle and medullary rays, can give rise to ground tissue system, whereas the procambium, which give rise to the vascular bundle, it will give vascular tissue system. Now let's check the intercalary meristem. This plant parts is considered as the apical part, where apical meristem is present, and this branch area is known as the intercalary meristem, whereas these areas can grow. The lateral parts are known as lateral meristem. The intercalary meristem, it occurs between the mature tissues. It also increases the height of the plant by increasing the nodal area of branches. If it is lateral meristem, then this meristem is also known as secondary meristem, as it can increase the girth or the diameter of the plant. Due to this, it is commonly observed in woody plants, especially the plant with mechanical strength, that is among dicots. Lateral meristem can develop into fascicular vascular cambium, interfascicular cambium and cork cambium. So this part we will see when we are going to discuss about the cambial ring activity. Now let's see the simple tissue under the permanent tissue. As already we had discussed that simple tissue can be divided into parenchyma, collenchyma and sclerenchyma. Now let's write some of the points for the parenchyma. The cells are isodiametric. It means all cells are equal in size. Number 2, the shape of the cells are spherical. It may be oval, round, polygonal or elongated. Its cell wall is thin and made up of cellulose. As you can see here we have written the intercellular space. So may have small intercellular space or intercellular space may absent. Under the collenchyma, we can see the structure. It has one thick corner, and this thick corner is because of deposition of cellulose, hemicellulose and pectin. So you can see here so many depositions are there at the corners of this kind of cells. It has a vacuole at the center and the cell wall is very thick because of deposition of these chemicals. Therefore, it has no intercellular space. The other character is its shape is oval, spherical and sometimes polygonal. Under sclerenchyma, as you can see here, we have two concepts. One is known as fiber and other one is known as sclerid. So the concept of sclerenchyma is that these are the dead tissues or dead cells, as they are that due to deposition of lignin. Its lumen is also very narrow and their cell wall is very thick. The fibers example are wood fiber and the bast fiber. Wood fiber is also known as xylem fiber, whereas the bast fiber is also known as phloem fiber. On the sclerids, we can see these are spherical, oval or cylindrical in shape. It has very narrow cavity. Now let's say the function of all the simple tissues. Depending on function, parenchyma is of different types. One is known as chlorenchyma. That means it has chlorophyll. These tissues are known as Palisade tissue and these are known as spongy tissue. The Palisade and spongy tissue together the Mesophyll Cell. So in the Palisade tissue we have chlorophyll and which can perform photosynthesis. Similarly, as it can do photosynthesis, it can store food as well. So this is the storage of starch. The spongy tissue, it has multiple intercellular space and these are actually the air cavities. So its cell name is parenchyma. Due to this air cavities, they provide buoyancy and present in aquatic plants. Similarly, the third function of the parenchyma is prosenchyma cell which develops the pericycle. I hope everyone understood the types at the same time the function of the parenchyma. Now, if we check the collenchyma tissue, its main function is to assimilate food. That means it can make food or can do photosynthesis as they have chlorophyll. The other function of collenchyma is it provides mechanical strength to young leaves and stems. On the sclerenchyma, the sclerids are present in fruit wall of nut, pulps of fruits like guava, pear and sapota, seed coat of legumes and leaves of tea. By understanding their position, it is very clear that it provides mechanical support to the plant. There is a difference between complex and simple tissue. Like in case of complex tissue, they are structurally and functionally different cells, whereas the simple tissues are group of cells which are structurally and functionally same. Complex tissue can be categorized into two different types, xylem and phloem. Xylem, it is the conducting tissue, that is, it carries water, minerals from root to stem and leaves. It also provides mechanical strength, whereas phloem is mainly for food transport mechanism, which carries food from leaves to the roots and other parts. So please remember here leaf is known as source and roots and other parts where there is no photosynthesis are considered as sink. Whereas opposite in the case of xylem, that is root is going to give all the minerals and the water to the leaves and the stem in case of xylem. Now let's check what are the elements present in xylem and the phloem. So xylem elements are of four different types: tracheids, vessels, xylem parenchyma and xylem fiber. On the tracheids, as you can see here, their ends are taper. Their lumen is narrower as it has thick wall. It is elongated cell and is dead due to absence of protoplasm. It is the characteristic feature of gymnosperm. On the vessels, as you can see, it is cylindrical. Its lumen is wider enough as the wall is thinner as compared to tracheids. It has some bordered pith, which is something like cavity. Vessel is a characteristic feature of angiosperm and absent in gymnosperm. On the xylem parenchyma, as the word indicates that it is the living tissue. Therefore, cell wall is made up of cellulose. It can store food such as oil, starch, and chemicals like tannin. Xylem fiber, as the name indicates, made up of sclerenchymatous tissue. It is highly lignified, therefore narrow lumen or obliterated lumen. It may have septa or without septa. Septa means partition. So suppose if it has this kind of cell, then it may have the partition like this or it may not have. So if it has partition, then this is known as septate fiber, or if it has no partition, then that is known as aseptate xylem fiber. So the common example is wood fiber. Now, depending on maturity, xylem is of two different types, protoxylem and metaxylem. If it is protoxylem, it forms first. As the name indicates, proto is actually for the first. If it is meta means big. So if it is big, it is lateral form. As it is first developed or newly developed, so it is known as immature xylem. And if it is later it develops, so it becomes matured enough. So it is mature xylem. The protoxylem, as it is first developed, so it has thin wall, whereas metaxylem, as it later develops, so it has thick wall as many deposition happens. So thin wall cells are always have small vessels as they are newly or very premature, and if it is lateral, so it is matured enough, so it's large vessels as well. Again, depending on position, xylem can be categorized into two different types. One is known as endarc and other one is known as exarch. Now, for example, let's take one stem section. If we cut at this particular side, we will get this kind of structure which has the metaxylem towards the periphery and protoxylem. These xylems are present towards the center. So this is known as endarc. End means inside and arc means this is the arc of the pith. So towards the pith, if protoxylem is present, then it is known as endarc because protoxylem is present inside. Similarly, if we check the exarch condition, here you can see the smaller size xylem, which are nothing but the protoxylem, and towards the center we have metaxylem. So as protoxylem is present outside, so X means outside or the outer region. So protoxylem is present towards the outer region or peripheral side and metaxylem is present towards the center or the pith. Therefore, this is known as exarch kind of xylem, which is commonly observed in case of roots. So please remember, in roots we have exarch condition, whereas in case of stem it is the endarc condition. Now let's move towards the phloem elements. So under phloem elements we have sieve tube, phloem parenchyma, companion cell and phloem fiber. So let's check one by one in brief. First one is sieve tube. So as you can see here, sieve tube is nothing but a long slender body. It is a cylindrical in shape. As you can see in this diagram, it has long and slender tube. Its cytoplasm is present towards the peripheral area as it has large central vacuole, but it has no nucleus. Interestingly, this is a question pocket that sieve plate and the sieve tube together, it is known as sieve tube element. So please remember the concept because the sieve tube element is there in your NCERT textbook diagram. So sieve tube element is nothing but the sieve plate and the sieve tube all together. Now let's check the companion cell. It has nucleus. As you can see in the companion cell, you can see some nucleuses are there. So these are the nucleus and this nucleus helps sieve tube to maintain pressure gradient. This is a characteristic feature of angiosperms. That means in case of gymnosperm, this we will not get. In gymnosperm, the Campanian cell which is present is known as albuminous cell. So this is one of the important information for the NEET examination. So please note it down, that is the albuminous cell is the characteristic feature of gymnosperm, and the same thing in case of angiosperm, we consider it as the Campanian cell. Remaining, as the name indicates, it is the parenchymatous tissue and it is living tissue which can store food materials along with resins, latex and mucilage. Please remember, it is absent in monocot. Again, the phloem fiber is the most important phloem component. It is also known as bast fiber, as already we have mentioned. Of course, if it is a fiber, then it is sclerenchymatous in origin and it is present in jute, flax, hemp, and all of these are used to make many economically important items like mat, shoe, rope, etc. So commercially it is very important. The bast fiber is very important, and this is one of the important question for the NEET examination. After completion of the complex tissue, now we need to see the tissue system. In that tissue system, we have epidermal tissue system, ground tissue system and vascular tissue system. Epidermal tissue system, it consists of epidermis, stomata and epidermal appendages. So if we consider the epidermis, that means we are considering the stem part. The same name we can give for the root part also, but in a different way. This is the epiblema for the root. In case of epidermis of stem and root, both are made up of parenchymatous cell. They are single layered. They have little amount of cytoplasm and vacuole is large. The difference between the epidermis and epiblema is that in case of epidermis, cuticle is present, whereas in case of epiblema, there is no cuticle. Now come to the stomata. This is the characteristic feature of epidermis. That means it is not present in epiblema. As you can see here, it may be bean-shaped structure or dumbbell-shaped structure. If it is bean-shaped structure, then this is the characteristic feature of dicot, and if it is dumbbell-shaped, and this is characteristic feature of monocot. The stomatal apparatus consists of stomatal aperture, which is nothing but the stomatal pore, the guard cell, which is consists of the chloroplast, and because of that, the guard cell can go for photosynthesis as well. Along with that, it has subsidiary cell. Please remember, stomatal apparatus is one of the important question for NEET examination. So it has subsidiary cell, guard cell and stomatal aperture or pore. As we know, stomata, the main function is to do gaseous exchange as well as transpiration. The inner lining of stomata is always thick and the outer lining of stomata is always thin. Please remember these two concepts. Inner one is thick and outer one is thin, which helps in stomata open and close. On the epidermal appendages, root has root hairs, which is unicellular. This is also one of the important concepts. Unicellular root hairs are present, and if it is stem, then it has trichomes. Trichomes means the stem hair which are multicellular in nature. If it is root hair, it absorbs water, and if it is stem hair, it checks loss of water. Interestingly, trichomes, it may be thorn, pine as well. I hope the epidermal tissue system is clear to everyone. Let's come towards the ground tissue system. Very simple concept. Leave the epidermal area and the vascular bundle, and in between these two areas, whatever we have like cortex, endodermis, pericycle, medullary rays, etc., including pith, all of them are considered as ground tissue system. It is made up of all types of tissues. That means it is made up of parenchyma, made up of collenchyma and sclerenchyma. Especially parenchymas are cortex, pericycle, pith and medullary rays are made up of parenchyma tissue in case of root and stem. If in case of leaf, the mesophyll cell contains thin-walled chloroplast containing ground tissue, and that we know as the Palisade tissue. Now the last part, and that is the vascular tissue system. On the vascular tissue system, we have two concepts. One is radial type and other one is conjoint type. If it is conjoint type or conjoint type, it means both the vascular bundle, that is xylem and phloem, are present in the same vascular bundle. And if it is radial, that means xylem and the phloem are present at different radius. As you can see here, xylem are present in this radius, whereas phloems are present on the alternate radius. So therefore, this type of condition of having vascular bundles is only and only available in case of roots. So root has radial type of vascular bundle, whereas in case of stem, leaves, it has conjoined type of vascular bundle. That means both the xylems are phloem and are present in the same radius. Some of them are known as cojoined closed and some are known as cojoined open. If it is closed, that means there is no cambium in between xylem and phloem. As you can see here in the diagram also, phloem and xylem, these two are separated but not with the cambium. So in this case, this is known as cojoined closed. In absence of cambium, it never can go for secondary growth. Monocot stem is the best example, whereas in case of cojoined open, it has cambium in between xylem and phloem. As you can see here, this structure is nothing but the cambium, and if cambium is present, that means it can go for secondary growth as well. So this structure is commonly observed in dicot stem. So this is the simplest way how we can remember all the parts of tissue system. After completion of tissue system, we can go for anatomy of dicotyledonous and monocotyledonous plant. The difference between dicot root and monocot root. In this case, the full structure is for both the dicots and monocot, but there is a simple difference if you check here, that this pit concept is only and only for the monocot. It is not for the dicots. So the root hair is present on the epiblema area for both dicot and monocot roots, which is followed by the cortex region. The cortex is parenchymatous in case of dicot, whereas in monocot it is the sclerenchymatous. But we can see in some cases parenchymatous as well. Then come to the endodermal area. In this endodermis, you can see two types of cells are available, in which in one's the suberin deposition. So those cells are known as Casparian strips. Whereas if there is no suberin deposition, then that is for the passage cell. So in the endodermal region, both the Casparian strips and passage cells are available. Next, pericyclar region is actually responsible to give rise to the cork and cork cambium only and only for the dicot. But in case of monocot, there is only the arisal of the lateral roots. So please remember, if it is the lateral root, its origin is always endogenous. So this is one of the important question pocket for the entrance examination, that lateral roots are endogenous or exogenous origin. In between the xylem and phloem, as you can see here, some cells are present and these cells are known as conjunctive. This is also one of the question pocket. So please note it down, this point also, that is the cells which are present between xylem and phloem. As you can see here, xylem and phloem, these two are actually separated. The red color marking is showing the phloem structure and the structure is showing the xylem behavior. So as you can see here, both the structures are present in the different radius. So the vascular bundle in case of roots of both dicot and monocot is nothing but the radial kind of vascular bundle. At the same time, if you check, the protoxylem is always present towards the peripheral region, whereas the metaxylem, which is the larger in size, it is present towards the pit or present towards the center. So this kind of xylem is known as exarch kind of xylem. There is a simple difference between the dicot root and monocot root with respect to the vascular bundle, that in case of dicot root, we have 2, 4 or 6 numbers of vascular bundle may present. That means they may develop diarch condition, tetrarch condition or hexarch condition. Whereas in case of monocot, more than 6 to 20 numbers of vascular bundles it may present. So their condition is known as polyarch. So depending on number of vascular bundle, dicots having diarch, tetrarch and hexarch condition, whereas in case of monocot it is the polyarch condition. Now let's see all these points how we can write in a stepwise manner. So here we have written all the information. You can see here number varies, like diarch, tetrarch, that condition also we have wrote. Here the polyarch condition we have written. Absent in case of like if it is cambium, then it is present only in case of dicot, and in case of monocot it is absent. Again, the pith is inconspicuous or small. That means you can say almost it is negligible, we can say, in case of dicot, whereas pith is well developed in case of monocot. So all the difference are written in this part. Hopefully this part will help you to understand and to remember all the points. After completion of this, let's check the next part, and that is the anatomy of dicot and monocot stem. So here we have drawn the structure for the dicot, whereas in this case we have drawn the structure for the monocot. Let us see their difference. Now you can see here, this is the cuticle layer which is present on top of the epidermis. Here also we have the cuticle layer on top of epidermis. That means the cell structure is same. But you can see here from the epidermal area, some outgrowth can comes out. So that outgrowth may be spine, it may be thorn, it may be hair, anything it may be. So it is known as trichome. The characteristics of having trichome is only and only for the dicot. The same trichome or the hair or any outgrowth like a spine, thorn, this kind of structure is not available in case of monocot. Hypodermis in case of dicot is made up of collenchymatous tissue, whereas in case of monocot the hypodermis is the sclerenchymatous tissue. You can see here that below this hypodermal area we have a distinct area for the cortex, distinct area for endodermis, pericycle, medullary rays and the vascular bundles. But here all the cells are almost same, therefore it is written as undifferentiated ground tissue. So from here we can easily describe that in case of dicot, ground tissues is differentiated into cortex, endodermis, pericycle, medullary rays as well as pith, and that pith is parenchymatous. Similarly, you can see here under the endodermal area it is something like wavy pattern. So it is wavy in nature, and here as there is no endodermis, so we can't see any kind of wavy nature. The pericycle here, it forms a cap. Please remember these terms are important for entrance. It forms a cap-like structure just above the vascular bundle, and if you see the vascular bundle, the first part is the phloem part. Then we have cambium in between the xylem and phloem, and the xylem, if you check, this is the metaxylem, and inside it we have the protoxylem. So this type of xylem is known as ender kind of xylem. So the vascular bundle in case of dicot is collateral, cojoint, or I can write conjoint also, and it is open. Open means where we can go for the secondary growth. So please remember, if it is open type, already we have the cambium, but here you can see that as this part is considered as the phloem and this part as considered is as the ender, that is xylem. So there will be no partition in between phloem and xylem, no cambium it means. So the vascular bundle is known as collateral, conjoint, at the same time it is closed, and closed means there will be no secondary growth as there is no cambium. So I hope this part is clear to all. Again you can see on top of the vascular bundle we have bundle sheet in case of monocot, whereas there is no such kind of bundle sheet on top of dicot vascular bundle. In case of dicot, the vascular bundles are arranged in ring-like fashion. In case of monocot, the vascular bundles are actually in the scattered manner. Now let's check what type of information we can write for this. Here we have described everything like epidermis in the dicot stem, single layered. In case of monocot also it is the single layered. So here main differences are there in the parenchymatous. Which one is the parenchymatous tissue? Which one is the sclerenchymatous tissue? Where is like bundle sheet is absent, where it is present. So these are some of the important notes that I have given here. So please write it down, and if there is any confusion, don't hesitate to write in the comment section below. After this, the anatomy of dicot and monocot stem. Now let's see the anatomy of leaf. In case of anatomy of leaf, we have again two more concepts, that is dicot leaf and the monocot leaf. The very basic idea about the dicot leaf is it has one broad kind of leaf-like structure, and if we check the monocot, it is narrower. In case of monocot, it has the parallel kind of variation, whereas in case of dicot leaf, it is reticulated venation. So the first part is the broad size leaf and here the narrow size leaf. Again, this is the second point as the reticulate venation and the parallel venation. Now you can see here this part, which is upper side, is also known as adaxial surface, and if it is the lower side of the leaf, then it is known as abaxial surface. So in case of stomata arrangement in dicot, it is known as hypostomatic. Meaning of hypostomatic is more number of stomata present in lower surface to check loss of water. Whereas for the monocot leaf, as it is isobilateral type of leaf, that means both the sides having the same amount of stomata, so it is also known as amphistomatic. That is, it has equal number of stomata on both sides of the leaf. Dicot leaf is also known as dorsiventral leaf. Now let's check the anatomy of dicot and monocot leaf. Let's start with the dicot anatomy. In case of dicot leaf, we have cuticle, and here also we can see the cuticle. So cuticle concept is for both dicot and monocot. Again, in case of upper epidermis, stomatas may present or may not present. That means as stomata is hypostomatic, that means maximum number of stomata present towards the lower side, so we may have or may not have stomata on the upper epidermis. But in case of monocot, we can see the stomata is present in equal number. So in both the upper and lower, we can see the stomata structure. So this is the difference between the dicot and monocot on the epidermal layer. Similarly, you can check here in the epidermis, we have some specialized cell, and these cells are also known as bulliform cell. Please remember, bulliform cell is also known as motor cell. The cell, it can retain water, and if it retains water, it becomes swollen, and in absence of water, it becomes shrink, and then the leaf curls. Therefore, bulliform cell help to check loss of water. Now, you can see here that this part is the mesophyll cell in between upper and lower epidermis, and the mesophyll cell is divided into two different parts. One is known as Palisade tissue and other one is known as spongy tissue. Both the Palisade tissue and the spongy tissue is chlorophyllous in nature. But in case of monocot, we have no such differentiation in the mesophyll cell. Whereas the vascular bundle, it has xylem out towards the upper epidermis and phloem is towards the lower epidermis. In case of leaf, xylem is always present towards the outer side. That means towards the epidermis. And if it is phloem, then it is present towards the inner side. Whereas this is exactly opposite for the stem and the root. This concept is for both dicot and monocot. So try to remember this question. This is also one of the important questions for NEET examination. Now you can see this xylem and phloem containing vascular bundle is actually supported by some type of cell which are known as collenchymatous. But here the cell which is present in the monocot, these are the sclerenchymatous, and it support the vascular bundle. The vascular bundle is same like enda kind of xylem, and the phloem is present below, and in between xylem and phloem there is no cambium. So please remember the vascular bundle for both dicot and monocot is collateral, cojoint and close tie. So this feature is common for both dicot and monocot. Similarly, you can see here that this vascular bundle which is present in this side, this vascular bundle is the young vascular bundle, and it is supported by some kind of parenchymatous tissue which is nothing but the part of mesophyll cell. Whereas in case of dicot, there is no such differentiation. For monocot, vascular bundle, if it is older, then it is supported by the sclerenchymatous, and if it is younger, then it is supported by the parenchymatous tissue. So these are the basic difference between dicot leaf and monocot leaf. Now let's check what are the different points for dicot and monocot we can write. So after seeing these points, I hope it is pretty much clear that whatever we have discussed in the diagram, the same points we have written here as well. After this, we have to go for the secondary growth of plant. In the secondary growth, it can be divided into lateral growth, which is also known as growth in the diameter, and growth in the girth. The lateral growth can be divided into two different types. One is known as vascular cambium and other one is known as cork cambium. The vascular cambium is also known as normal kind of growth, and the cork cambium is also known as anomalous secondary growth. The vascular cambium or the normal secondary growth is developed into the fascicular cambium, whereas the anomalous growth is developed into phallogen, phloem and phalloderm. Under the vascular cambium or the fascicular cambium development, towards the peripheral side, phloem is usually present, whereas towards the inner side, xylem is present. To separate the xylem and phloem, we have one more structure, and that is nothing but the cambium, intrafascicular cambium. If different vascular bundles are present, and in that vascular bundle, the cambium ring or the intrafascicular cambium ring, it can join together and can make another structure which is known as interfascicular cambium, which can connect all vascular bundles. In the vascular region, if there are so many vascular bundles, then all the vascular bundles are linked together with the help of the interfascicular cambium and the intrafascicular cambium, forming a ring-like structure known as cambial ring. After formation of cambial ring, now let's check what is the activity of cambial ring in the secondary growth. As we can see here, the intrafascicular cambium towards the outer side, that means where there is phloem, it can develop some layer, whereas in the inner side also it is very active where there is xylem. But towards the inner side, the intrafascicular cambium is more active as compared to the outer side. The primary xylem, which is present towards the inner side, is pushed by the activity of the intrafascicular cambium, which makes a layers of secondary xylem. The activity is too much, because of which the deposition is going to develop multiple layers of secondary xylem, which can push the primary xylem. Whereas towards the outer side also, the secondary phloem is developed due to the deposition of intrafascicular cambium. As the activity is too much towards the inner side, the primary xylem is going to push the pith. Therefore, the cells of the pith area is crushed with the primary xylem, and when the vascular bundle become older enough, the pith become narrower in the dicot stem. Therefore, in dicot, the pith is known as small, narrow, less or inconspicuous, whereas the secondary phloem is also going to push primary phloem, which pushes the pericycle. This case happens when the plant is older enough. After completion of the normal growth, let's check the abnormal or anomalous secondary growth in the dicot stem. Below the endodermal layer, the vascular bundle is present where xylem and phloem are separated by the cambium. Above the vascular bundle, the pericyclar layer, which is behaving like parenchymatous in nature, it can develop phallogen by depositing some kind of substances. The phallogen is also known as cork cambium and it is parenchymatous, due to which can deposit suberin, which make the cell hard and known as cork or phloem. Due to deposition of suberin, this layer is water resistant. Now come to the inner side where phallogen can deposit different types of chemical substance which can make phalloderm, or it is also known as secondary cortex. The suberin deposition and other chemical deposition is too much towards the cork or phloem area, which ultimately results into cell bursting. Therefore, the cork is hard and the phalloderm is soft. Together, phloem, phloem and phloderm is known as periderm. Bark of the tree is actually comprises of periderm and the secondary phloem, which is present on the outermost surface sides of the plant. Sometimes in that bark, lenticels are present which are responsible to exchange the gases. So one should remember the terms like bark, periderm, secondary xylem, functions of lenticels, etc. These questions are important for NEET examination. Please remember this bark, periderm, lenticels are the characteristic feature of woody plant, or it is the characteristic feature of dicot plants. The last part of the secondary growth in the stem part is formation of spring wood, autumn wood, sapwood and heartwood. Spring wood and autumn wood is determined the age of the plant as it can give the annulus ring. Annulus ring can be developed during the spring season and autumn season. For that, here I am writing one question, and that question you have to solve. After solving, please write in the comment section below what is the answer of this question. Let me give you one hint here. Within a year, spring wood and autumn wood both develops. Now come to the sapwood and heartwood. If it is sapwood, it can store water and minerals. Because of deposition of water, it is not durable. Whereas in case of heartwood, it deposits lignins wherein waxes, tannins and resins. Therefore, it is durable and it is dark as well. In case of spring wood, the color is also light due to deposition of food and the water. Whereas in the autumn wood, the color is become dark because of an unfavorable condition, food and water cannot be deposited. So there will be deposition of different substances which make the color darker. The last part of this topic is a secondary growth in roots. From here, we need to understand the 2 points, and these are in case of angiosperm and gymnosperm, secondary growth can be observed in root and stem both. But though we have written angiosperm here, exactly the answer is for the dicot, because angiosperm it has monocot as well, in which secondary growth cannot be observed in stem and the roots. I hope the part of the anatomy is very clear to you people, and it is only one shot go. So we have described almost all the points that we needed for the entrance examination. Please study well, and if you have any confusion, you write your doubts or questions in the comment section below. Don't forget to subscribe. Thank you so much.